Okay, very welcome back to all of my dear viewers and obviously my friends and my student. So today's session I am going to discuss a very important topic of forensic medicine, and the name of topic is liver motis. So this liver motis, they are also known by some other names. We also call it as a post-mortem staining, post-mortem hypostasis or lividity. So they are some other name of this liver motis. So first we try to understand what is this liver motis. So we start the topic with definition of liver motis. Okay, what is liver motis? So liver motis is a early post-mortem changes. First point is liver motis is the early post-mortem changes, and this early post-mortem changes now they occur in the dependent part of the body which is not touches the ground. So here there are two keywords. One is the dependent parts of the body which is not touches the ground. So what is the meaning of this dependent part of the body? Dependent part of the body means the part of body which is faced towards the ground. For example, if the body is in prone position, then our front part of the body they faces towards the ground. So these are no, they are dependent part of the body. While if the body in supine position, then back of the body are the dependent part of the body. The second important point is that this dependent parts of the body they not touches the ground, because if they touches the ground now there is not any possibility of the stasis of the bloods, because the blood's capillaries are in compressed stage. So if the blood capillaries are in compressed stages, how there is possibility of a stasis? A stasis is only possible if the blood's capillaries is dilated. If the blood's capillaries is already constricted, then no any possibility of a hypostasis. So in the pressure area, there is the compression of the blood's capillaries, so no any possibility of pulling or the stasis of blood. So this liver motis are not occurring in those dependent part of the body which is touches the ground or a pressure is applied over it. In that area there develops a contact flattening. So contact flattening is basically a paleness area due to the pressure applied. So what are the other things in liver motis? Basically what are the different color changes, or normally in the liver motis there is bluish purplish or the purplish reddish appearance that you appreciate in the dependent part of the body. So now the question is arise, why this blue or purplish or reddish appearance will occur? This is because of the pooling of the bloods. When a person is dying, there is the cessation of the CVS. So if heart fail to pump the bloods, there is the stasis of bloods in the dependent part of the capillaries, and basically it is due to the RBC, and due to the stasis of RBC in the dependent part of the capillary, a bluish, purplish or the reddish appearance is appear. We call it as postmortem staining. So this is all about the postmortem staining. So now the time of appearance of postmortem staining, it appear after 30 minutes of the death, or I mean somatic death, and it is fully develops within 4 to 5 hours. So within 4 to 5 hours, this bluish or purplish appearance is fully develops. Now one more important point related to this postmortem hypostasis is what is the fixation. So what happens in case of the fixation? First we try to understand how many times take for the fixation. So fixation generally occur after 6 to 8 hours of somatic death. So after when how we decide a dead body, I mean post-mortem staining is fixated or not, we just gently press that post-mortem staining area. If we observe any color change, that means no, there is no fixation occur in the post-mortem staining. But if if there is no color change, that means fixation is occur, and if there is a color change, that means fixation is not occur. If there is fixation occur, that means the death of bodies is occur more than 6 to 8 hours, and if the fixation is not occur, that means the body death will occur less than 6 hours. So these are one of the medical legal importance of the fixation. On this basis we find what are the time since death. So now the question is arise, why this fixation occur? This fixation is occur because after the death of the person, vascular permeability is increases. So when the vascular permeability is increases, there is the infiltration of surrounding tissue by RBC. Normally RBC are present inside the blood vessels. Now, they are not infiltrated into the tissue. But after the death, the vascular permeability is increases. So due to the increase of the vascular permeability, now the RBC infiltrate the surrounding tissue. And when the RBC infiltrate the surrounding tissue, now they cause permanent fixation. That means no any color changes when we press the post-mortem staining area. So this is the mechanism of fixation. Now we go for the reading section. We start with the definition or the livor mortis. They also known as the post-mortem staining, post-mortem hypostasis, and the lividity. Okay, definition: it is the early post-mortem changes occur in the dependent part of the body which not touches the ground. One important point: dependent part of the body not touches the ground. Appear bluish purple or purplish green due to the pooling of bloods in the capillaries of the dependent part of the body. Now another definition is the contact flattenings. So it is the paleness of that dependent part of the body which in contact of the ground or the pressure applied over it. Okay. Time of appearance: it began after the 30 minutes of the somatic death and the fully developed within 4 hours. What is fixation? So fixation is time developed within 6 to 8 hours. Now we go for a discussion of different types of color changes which indicate different cause of death. So first is, if the post-mortem staining color is the cherry red, it indicates the carbon monoxide poisoning. If the post-mortem staining is green color, hydrogen sulfide poisonings. Brown color, yellow phosphorus. Chocolate color, aniline and nitrates. Black color, opioid poisonings. Bright pinks, hypothermia, and the bright red, that means hydrogen cyanide poisoning. So what are the medical legal importance? The first medical legal importance is the time since death depending upon the fixation. That means if fixation is not occur, it means the body is die, not time since death is less than 6 hours. If fixation is occur, then that means the time is more than 6 to 8 hours. Cause of death depending upon the different color, I already explained here. And here the differential diagnosis is the congestion and the contusion. Differential diagnosis means basically they resemble to the post-mortem staining, but actually they are not a post-mortem staining. One more important word is the vivaces. What is vivaces? Vivaces is the bluish purple tiny hemorrhages appear over the post-mortem staining area. So sometimes what happens in the post-mortem staining area, there is the appearance of tiny bluish purple dots like appearance called the vivaces. Okay. So this is all about the post-mortem stainings and the liver mortis. Welcome back in new lecture.